The very first thing you should know about biology is that it's the study of life. But honestly, that's not quite the whole story. Life is just a word we use to explain a bunch of different molecules and chemical reactions that come together in incredible and unexpected ways. Now, when we piece all of these molecules together, sugars, fats, proteins, somehow we end up with a living cell. And living cells are some really incredible things. They're organized, they grow, they develop and reproduce, they maintain a constant state of internal conditions, they respond to stimuli, and they have a heritable genetic code that allows for adaptation and evolution. These walruses, for example, are alive, but the iceberg behind them is not. Interesting. Biology is a discipline of science, and science refers to the knowledge we have about the world and universe around us and the logic and reasoning that we use to obtain this knowledge. Sciences that relate to the Earth and stars are called natural sciences, and they're divided into 5 major disciplines: astronomy, biology, chemistry, earth sciences like geology, climatology, and hydrology, or water science, and finally, physics. We can also categorize the natural sciences as life sciences, or physical sciences. Life sciences focus on the biotic living stuff, and the physical sciences focus on the abiotic or non-living stuff. So, biology, being the study of life, can clearly be classified as a life science. But what about the other 4 natural sciences? This is where it gets confusing. Astronomy, for example, is the study of galaxies, stars, planets, and other celestial objects. Astronomers often use mathematics, chemistry, and physics to understand how these astronomy things are formed and how they interact. Therefore, can we safely refer to astronomy as a physical science? Or can we? Some astronomers study astrobiology by applying the basic principles of biology and biochemistry as they search for extraterrestrial life or for potentially habitable planets based on our understanding of Earth science. This tells us that the 5 disciplines within the natural sciences are not mutually exclusive, but in order to study complex phenomena, we often need to take an interdisciplinary approach. Okay, back to biology, the study of life. Life, though, can be surprisingly difficult to define. For example, if you've ever visited an aquarium, you probably noticed that jellyfish don't seem all that different from a plastic bag floating in the water. Both are thin, semi-transparent, and sort of move with the current. Yet jellyfish are most definitely alive. Life is defined by a set of characteristics that are shared by all living organisms. I've seen some longer lists out there, but there are really only 5 minimum requirements for life: cellular organization, response to stimuli, growth, development, and reproduction, which are all interrelated concepts, homeostasis, and evolutionary adaptation. Let's run through this checklist with another animal. We can confidently say that this danger noodle is alive because it consists of 1 or more membrane-bound cells, and each cell contains complex molecules, can detect and react to external stimuli or changes in its environment, goes through regulated and coordinated processes to increase in size, change or mature, and or reproduce, maintain a constant state of internal conditions different from its outside environment, this is called homeostasis, and has a heritable genetic code that allows for adaptation and evolution. Organisms use large amounts of energy in order to do what they do. Even evolutionary adaptation requires energy because the organism's genetic code must be maintained and replicated during the process. All the ways that an organism collects and uses energy is referred to as its metabolism. In biology, everything has a hierarchical organization, and higher levels build on lower levels. For example, the human circulatory system is composed of the heart and blood vessels, which themselves are composed of muscle tissue and connective tissue, including blood. And those tissues are made of different types of cells, and so on. There are a lot of different levels in biology that span the cellular, organismal, and ecological scales. We can start at the level of atoms, building up to molecules, macromolecules, organelles, and cells. Then, at the organismal scale, we deal with tissues, organs, organ systems, and the whole organism. Finally, at an ecological scale, we see populations, communities, ecosystems, landscapes, geographic regions, biomes, and the entire biosphere. At the ecological scale and above, we're also going to place an equal emphasis on abiotic factors. For example, sand, soil, water, trees, shrubs, hoofed mammals, and humans are all important parts of this river ecosystem in Nigeria. We can't always predict what will happen at higher levels of organization based on what we know about the lower levels. Complex interactions can often lead to completely new phenomenon called emergent properties, like how the behavior of a top predator might affect an entire community of animals in an ecosystem. Biology might be the study of life, but it's rooted in the physical sciences of chemistry and physics. Below the level of the cell, it's nearly impossible to predict how proteins, nucleic acids, and nutrients might come together in such a way that meets all 5 of the key characteristics in our definition of life. This means that life itself is an emergent property of atoms and molecules. As early as the age of the philosophers in ancient Greece, people have recognized that life can be placed into one of 2 kingdoms, plant or animal. Since that time, other systems have arisen to explain the classification of living organisms. In 1866, Ernst Haeckel suggested a 3rd kingdom, the protists, or kingdom protista, which included a wide variety of unicellular organisms like bacteria. In 1969, the plant ecologist R.H. Whitaker proposed a system that differentiated between prokaryotes, organisms with cells lacking a nucleus, and eukaryotes, organisms with nucleated cells. Whitaker's system recognized 5 kingdoms: Animalia, Plantae, Fungi, Protista, and Monera. The kingdom Monera included all prokaryotic organisms. Now, these systems of classifying life into kingdoms were largely based on cell type, food source, and mode of reproduction. But organizational systems are anthropocentric. They were created by humans for the purpose of making sense out of the world that surrounds us. More recently, the availability of molecular data has revolutionized how we classify living things. In a new system devised by Carl Woese and colleagues, we now use molecular data to divide all living organisms into one of 3 domains: bacteria, archaea, or eukaryota, or eukarya. Domains sit above the level of kingdom, and plants, animals, and fungi are still kingdoms in the domain eukaryota. Monera, the prokaryotes, were divided into the domain bacteria, which contains all true bacteria, and archaea, which differ significantly in the structures of their membranes and ribosomes, among some other factors. Sadly, the kingdom Protista, which would be eukaryotic, is considered invalid since it's paraphyletic. Animals, plants, and fungi all evolved from protists. Taxonomy refers to the naming and classification of species using hierarchical ranks, and traditional taxonomic ranks were developed by Carl Linnaeus, a Swedish botanist and zoologist. Linnaeus grouped species based on similarities in their morphology or anatomical structure. His organizational system forms a nested hierarchy, with kingdom being the highest level. Well, domain was added recently, remember? Followed by phylum, class, order, family, genus, and species. Linnaeus also developed the system of binomial nomenclature to name species using a unique combination of two words. The first word of a species is its genus, and the second word is the species epithet. A genus references a specific group of organisms, so the name of a genus can only be used once, although there are exceptions, like how a genus can be used once for a group of animals and again for a group of plants. And species epithets are adjectives, so while a single epithet can only be used once per genus, they can be repeated across many different genera. When writing the name of an organism, like our own species, Homo sapiens, both the genus and species epithet should be in italics, and the genus, but not the species epithet, should be capitalized. Many people believe the ultimate goal of science is to know. At first glance, it sounds like this is an extremely grand venture, but it's actually a bit of a selfish perspective, because the usefulness of science is to solve problems and improve our quality of life and the health of the environment around us. Knowledge alone is worth relatively little. It's our actions and how we use that knowledge that have the greatest effect. Curiosity and creativity are valuable traits for any scientist, and at the most basic level, science is about looking at the world around us and wondering why things are the way they are and what might happen if something were to change. While there's no single correct way of doing science, the scientific method refers to the basic steps used for carrying out experiments and evaluating results. The scientific method is a circular, repetitive process, not a linear one. Every step of the scientific method is important, but scientists might approach each step differently depending on their field and study system. Regardless, the end goal is to make sure that what you're doing is sound science. When we talk about the scientific method, there are a few key elements beyond this whole observe, research, hypothesize, test, analyze, and report cycle. First, that scientific phenomena are subject to cause and effect. If I drop a rock, it falls to the earth. If I touch a hot stove, my hand gets burned. This ties in with another theme. The scientific process should be reproducible. It shouldn't matter whether I drop a rock today or tomorrow, it should fall to the earth every time. Likewise, our hypotheses need to be testable. Otherwise, it's just an invalid premise argument. As an example, let's hypothesize that the weather is controlled by telepathic dolphins living in castles in the sky. Unfortunately, there's no way to experiment with the sky dolphins, or even find them. So this hypothesis is not testable and cannot be addressed using the scientific method. Okay, second, a hypothesis needs to be falsifiable. We should be able to collect data that can prove it wrong. For example, if someone claims that Bill Gates is secretly using the COVID vaccine to implant us with microchips, but these microchips are too small to detect, then this isn't falsifiable. There's no possible way to collect evidence to reject that claim. We use the scientific method in all fields of biology. For example, think about a patient coming into an emergency room with chest pain. We're worried that this patient might have had a heart attack and needs treatment right away. We'll run a few tests, including an electrocardiogram, ECG, to look at the electrical activity of the heart, as well as a blood test to check for protein markers associated with damaged heart muscle. In this case, both tests come back negative, so we can say with a very high level of confidence that the patient did not have a heart attack. It's not 100%, but it's about as close to that as we can get. This means we can safely rule that out while we continue to run other tests. Albert Einstein once said, "No amount of experimentation can ever prove me right. A single experiment can prove me wrong." The scientific method is a powerful tool for conducting sound science. By using the scientific method, we're acknowledging that it's the process that makes something science, not the results. Science isn't about getting a certain answer, but the way we go about it. Einstein has also said that so many people today, and even professional scientists, seem like somebody who's seen thousands of trees but has never seen a forest. And this brings us to another common misconception about science, that it's the simple pursuit of knowledge. Really, science is about the broader context and applications of this knowledge. Regardless, science is not a belief system. There are no absolute truths in science, only differences in the level of uncertainty. I'll talk about that more later in this video. When we apply the scientific method, we're using logic and reasoning to make inferences or reach conclusions. The reasoning we use can be deductive or inductive. Deductive reasoning applies a general principle to a specific situation. For example, take the premise, all animals with hair are mammals. If I pick an animal at random, and my animal has hair, then I can infer that this animal is a mammal. But if my animal doesn't have hair, does this mean it's not a mammal? Or is the argument invalid, which means something totally different? Think about it. The opposite of deductive reasoning is inductive reasoning. What we do here is take specific observations, put them together, and use them to form broader principles. So let's say I go to the animal shelter looking at all the cute puppies that need a home. I noticed that terriers, retrievers, Pomeranians, and Shiba Inu all have hair. I might use this information to conclude that all dogs have hair. We've already talked about how a phenomenon needs to be subject to cause and effect and reproducible, and how a hypothesis must be testable and falsifiable in order for us to address it through the scientific method. So, what is a hypothesis? A hypothesis is a proposed explanation for a certain set of observations. It fits the facts to the best of our knowledge. Once falsified, or if falsified, we can reject it in favor of some other, better hypothesis. A hypothesis must be able to generate predictions, and it should be testable and falsifiable. Experiments and observations gather data that may or may not support a hypothesis. If the data support our hypothesis, then we can accept it, for now at least. But if the data don't support our hypothesis, then we may modify or reject it completely. In science, it's rare to limit yourself to a single hypothesis. There may be several possible relationships, interactions, or other mechanisms at play, and we'd like to know what's really going on. Sometimes there's no relationship between one variable and another. For example, if I turn on a light in my bathroom, then this will have no effect on the light in my bedroom, because the switches are independent of each other. A hypothesis of no effect is called a null hypothesis, or H0 or HO for short. But when we test something, it's usually because we have reason to believe there's a relationship between the variables. We expect to reject the null hypothesis in favor of some other hypothesis. Maybe I suspect that the wiring in my house has water damage, like there have been some heavy rains and I've noticed flickering lights in the past. So my alternative hypothesis, or HA, could be if I turn on the light in my bathroom, then lights in my bedroom will flicker on and off because water has damaged the wiring. We can have more than one alternative hypothesis. If that's the case, we'll use the abbreviations H1, H2, and so on. Maybe the flickering lights I've noticed in the past are due to faulty wiring, not water damage. In that case, we might expect to see the lights in the other room simply turn off. If I turn on the light in my bathroom, then the lights in my bedroom will turn off due to faulty wiring. One final point. A lot of people are going to tell you that a hypothesis has an if-then format. Usually, this is because at least one prediction is already clear within the hypothesis itself. But personally, I'm not a fan of this format because it's almost always missing an explanation for the relationship between variables. Notice that when I wrote hypotheses about my flickering lights, I always included the mechanism within the hypothesis. Sometimes a variable doesn't affect anything else. This is called a negative result because we weren't able to reject our null hypothesis. Unfortunately, many scientists consider negative results to be a sort of failure, which means they won't share this with other scientists, or, as peer reviewers, will actively prevent other scientists from publishing negative results. But really, it's important to publish our negative results so other scientists don't keep testing the same things over and over. Repeatability is good, but we need to be doing that purposefully, not just because another lab decided not to share their results. And well, if the negative result was unexpected, then this could mean there's something else going on we haven't even considered, which itself is very interesting. When we test a hypothesis, we're not actually testing that hypothesis directly. We test the predictions that were associated with it. And remember, this process is based on deductive reasoning. When we use deductive reasoning to make an inference earlier in this video, we assume that the original premise was true, but we don't know for sure if a hypothesis is true. For example, if we hypothesize that all dogs have fur, we would predict that if we start picking dogs at random, then they will all have fur. I don't really have a specific mechanism for this. Besides, maybe they have fur because they're mammals. Anyway, this is testable, and we've used deductive reasoning by going from general, all dogs, to specific, this dog. And the more dogs we look at, the more support we might get for our hypothesis. Who's up for a trip to the pet store? But where did our hypothesis come from? We formulate hypotheses based on past observations and existing knowledge. Maybe we started out by watching the dogs in our neighborhood. We noticed that terriers, greyhounds, and poodles all have fur. Next, we apply inductive reasoning to conclude that all dogs have fur. In this case, we go from specific neighborhood dogs to general, all dogs. The natural world is affected by many variables, but in an experiment, we only change one variable at a time. The variable that we change is called the independent variable, and in response, we measure a different variable, one that we think will be affected by that change. This response variable is also called a dependent variable because whatever happens to it depends on the change we made before. In our control group, we leave everything unchanged or use a placebo. Basically, this is the default setting. But in the test group, we change the independent variable. And by comparing the results from the control and test groups, we can learn about the potential relationship between the independent and dependent variables. Of course, we can't trust the results of our experiment if we're only testing one or two different individuals. For example, if we were testing the effects of a fertilizer on plant growth, we'd probably need at least 20 plants in our control group and another 20 in the test group. We use the term replication to say how many times we repeat the different conditions in our experiment. Why do we need replication, though? If there's a relationship between two variables, should the results be obvious right away? Well, uncertainty comes from natural variation, and it also comes from exactly how precise we are when we're making our measurements and calculating averages. We need replication so we can isolate the effects of our variable from the randomness and uncertainty of the world around us. Observations and experiments are one approach for conducting science. Models are another, and they're particularly well suited for integrating what we know and generalizing results. We can use models to break down complex systems into their individual components, a type of reductionism, understand the effects of variables we can't manipulate or would take years to study, make short-term predictions or long-term predictions with greater levels of uncertainty. There are many topics in biology that frequently use models, like studies of cell signaling, protein structure, metabolic pathways, population dynamics, species distributions, and climate change. There are also applied fields that use models, like bioengineering or bio-inspired modeling. For example, the aerodynamic design of Shinkansen bullet trains in Japan was modeled after the bill of a kingfisher. I'm going to say this a lot. The scientific method does not prove anything. It's about collecting evidence that we can use to explain a process or phenomenon. This means that through the scientific method, we can only support or reject a hypothesis. Scientists can reject a hypothesis if it isn't supported, but rejecting a hypothesis is not really the same as proving it wrong or disproving it. There's always a chance of a false positive, also called a type 1 error in statistics speech, which means we rejected the null hypothesis after finding support for the alternative when the null was actually true. As scientists, we carefully design our experiments and use statistics to account for uncertainty, to bring uncertainty as close to zero as possible. During the report conclusions step of the scientific method, we also consider the possible limitations of our experiment and place our results in the broader context of the scientific literature. And when we report our conclusions, we also need to be clear about our methods, so other scientists can reproduce or build on our work. Science is not about getting the right answer, but about asking the right questions. There are no absolute truths in science, only differences in the level of uncertainty. Uncertainty can come about in many different ways, including the precision of measurement, accuracy in calculating the mean, existing natural variations, heterogeneity, and diversity. This can have important consequences, when it comes to predicting or forecasting farther and farther into the future, which definitely comes with increasing uncertainty. So if there's no proving anything with science, why do scientists consider theories to be as good as fact? To the general public, a theory is just a guess, right? It's just a theory. But scientists clearly use this word differently. To scientists, a theory refers to an accepted principle or body of knowledge made up of interconnected concepts, hypotheses that have been supported time and time again by scientific reasoning and experimentation. In the field of biology, our important theories include cell theory and the theory of evolution. A theory itself is a framework for organizing knowledge, and they're supported by a significant amount of evidence. So really, around 95% plus of studies should be supportive. Of course, a lot of theories are quite complicated, so the specific conditions and assumptions of each experiment can lead to results that might seem contradictory. Human error and random chance can also produce conflicting results, and these are all reasons why some theories are considered controversial and frequently argued about, at least by the general public. The best advice I have for you is to stay objective and consider all of the evidence and the different sources that this evidence might come from. All right, well, that's about it for today's video. Be sure to like and subscribe if this helps you study or just gives you a bit more insight into the science of biology. As always, thanks for watching, and if you have any questions, leave a comment or feel free to reach out in any way.